So good morning, everyone. So I will have a short presentation. There are a couple of topics I just want you guys to know about, and then I think there is an exam. It's a multiple choice, and there is, I think, one question where you have to write something, short answer. So I mentioned many times in my lecture that microbiomes are becoming more and more important and relevant, not just in cancer, but almost every day in our life. These are microbes which are residing in our body. I think they are more in numbers compared to what cell number we have. I think 3 times more, or 1.3 times. Three times more than number of cells we have, okay? So there are more microbes than our own cells. And those microbes basically are not just there as a bystander. They are basically critical for our, physiology, normal physiology. And they do impact many of our physiological activities and play important role in pathology. So in immunity also it has been shown that microbes are becoming more and more relevant from diagnosis to therapy. So microbiome is basically at this moment at a very crossroad between our physiology and pathology, not just in cancer actually, in every other disease we can think about. The intestinal microbiota plays a crucial role in the lifelong programming of innate and acquired immune response. And that's something you can take home message type thing, that you need to remember that these are there and they play a major role in both innate and adaptive immune response. And their main function basically is that fine tuning which involves a very precise and effective immune response is maintained, or actually is that balance which going from immunosuppressive to pro-inflammatory and all those kind of things are maintained more or less by the microbes which reside in our intestine. Several therapeutic modalities could be harnessed to restore the homeostasis of the gut, and this has been exploited not for our day-to-day living, like if you really leave and go for some other country or other part of India, I will suggest that take some probiotic before you leave. While you are there, keep taking it, and when you come back, take it, okay? And they are available. And before I left US, my doctor basically gave me this travel kit, which had probiotic and also doxycycline. So I have to start taking probiotic 1 week in advance, continue taking while I'm here, and I will have to take when I go back for 1 week. And doxycycline, if really I get sick, so just take doxycycline for 1 week. So that's like a kit they give you if you're coming to India. If you're going to Africa, there will be a different kit which involves other malarial drugs also in that, or vaccination for yellow fever and so on. So I think for every country, but all in all, coming back to that, microbes really are very essential for our physiology, and they are turning out to be important in pathology too. And some of the therapies, when you look at, and if you go and look at the published data, they are clearly showing that depending on your microbiome, the response to chemotherapy could be different. And this chemotherapy, just you are looking into plain and simple chemotherapy to most commonly used chemotherapeutic drugs. So the variability among different individuals could be, assigned to the type of microbiome one has. Another aspect is that when you are going through this, destructive chemotherapy or radiation therapy, microbiota is the first thing which gets destabilized, and that also can, relate to that toxicity or the abnormal, responses which they have. Microbiome in the gut basically is important in that role, and that, as I mentioned, they are very much involved in regulating our immune response. And this is getting more and more harnessed in terms of cancer management, because if you can maintain the normal microbiota, probably you can have a better response, not just to the chemotherapy, but all the immunotherapies which will follow that chemotherapy. And here is a very simple cartoon. The therapy can be optimized based on your microbiota. The efficacy of that particular therapy, the toxicity which is associated with that therapy could be tied to the microbiota, and those kind of studies actually have been published, and you can find lots of reports to suggest that those toxicity and efficacy of particular cancer therapy is related to the microbiome. It can be used as a biomarker depending on the response. So if you have such and such species, in microbiome, probably you will be able to predict particular type of therapy which will be more effective. Microbiome can be used as therapy itself. If it turns out that the cocktail of 6 or 7 bacteria probably enhances therapeutic response, you can create that cocktail and use that in enhancing the type of therapy. And it's not a dream anymore. These kind of work is ongoing right now, clinically as well as in preclinical models. And it can actually also, like everything else, big data and bioinformatics is helping in trying to find out those clones or those clusters which will be useful in that kind of therapy. And of course, at the end of the day, probably some of the products which are produced by these bacteria or microbes will be useful in generating therapies which will be more targeted towards the microbiome, modifying their responses and enhancing the therapeutic outcome, or limiting the toxicity. Toxicity in cancer therapy is a major concern. So if you can limit the toxicity, probably you can increase the therapeutics by just protecting those individuals. So all in all, there are a lot of big words in this slide, but all those big words are worth it to remember if you can. The complex interplay between cancer immunity and microbiota may be partially elucidated by novel omics technologies such as metagenomics, metatranscriptomics, culturomics, metabolomics, co-occurrence analysis, and three-dimensional crypt stem cell-derived enteroids in contact or distinct species of microbiome and immune subset, all integrated through computational biology. So all the omics you can think about are getting utilized in these interactions. You can imagine that you are basically, they have to create these models, using microbes, cells, cancer, and therapeutics which they are using it. So there are many, players which they get involved into, and each one of them are integral to outcome, But at the end of the day, once they can identify, that a particular, species, a particular group of microbes are critical, are helpful, they can create that kind of cocktail and will be helpful. Such an apothecary of omics technology will generate diagnosis tools for dysbiosis, for drug screening and novel therapeutics, such as artificial ecosystem or new microbial species, as well as small bioactives that either act on the microbiota to induce favorable shift in its composition or mimic desirable microbial effect to the host to enhance the overall outcome. So basically, at the end of the day, there are a lot of outcomes which will be created, but I like the word artificial ecosystem, that you all are aware of the ecosystem. These microbes within our intestine are more or less functioning in that kind of ecosystem, where there are factors produced by us helping those microbes, factors produced by microbes helping the host, and that interaction keeps going. The interventions in cancer therapeutics include prebiotic, probiotic, live bacteria are associated phases, natural products, autologous or allogenic fecal, microbial, transplantation. Okay, so there will be time will come when the people will start selling their stool. Okay, it's not a joke. You can buy it on Amazon even right now for allergy treatment. It's available on Amazon. Not for cancer treatment, by the way, but I think there is a fair possibility that this is going to happen. Pre and probiotics are already in market. Live bacteria we eat every day. When you are taking yogurt, that's what you are doing. Natural products like these fecal materials. I think maybe one day will come, and not to eat, but there will be some transplant method to transplant them from one patient to another. And it's all actually happening. And if you look and go in the literature and try to look for utilization of microbiome in cancer management, you will find lots of publication. I will say thousands of publication, not few. Okay, so this area is very hot. A lot of people are working. These interventions will have to be adapted according to the patient lifestyle, comorbidity, co-medicines which they are taking, and the genetic inheritance for an optimized personalization of his her therapy, which is possible in this kind of scenario. When you're talking about this, when you can create these microbiomes, you are basically personalizing overall outcome. So it is very much possibility that this will be very personalized rather than a cocktail which is available or which companies will sell as a medicine. Monitoring of microbiome will also be essential as a pharmacokinetic and dynamic parameter longitudinally in new interventional study that will be aimed to target the intestinal ecosystem. In cancer, whatever they are trying to do, a lot of studies have been done in other gut-related diseases or disorders, and actually cancer is just trying to exploit whatever has been learned. And the best example is your IBD, IBD where microbes play a major role in creating those inflammatory milieu. So actually microbes have been utilized in treatment of IBD, inflammatory bowel disease. There is no medicine for that, and I think microbiome is really helping them in conserving their microflora and microbiota. So all in all, that's a microbiome and cancer I had, and I will switch gear to now go into this next few slides talking about the revolution in immunotherapy and giving us just some example about what has happened in last 2 decades. And I hope you can really appreciate that. And more or less, I think this is a cartoon which was published last year in Science, where basically they were talking about those personalization of immunotherapies. All those mutanome-based vaccine strategies which I discussed with you yesterday, this was the cover of that review article which was published. So there is a fair possibility that new generation of cancer immunotherapies will include ways to engineer patients' own immune cells to kill their cancer, and patient-specific new antigen vaccines will be a very much possibility. So that's basically the day will come when these vaccines or these therapeutics will be very much tailored to the type of patient, not just a generalized treatment plan. So if you look at overall the immunotherapy, what has happened in last 20 years, a lot of studies very early on, and we are looking into this 1994 to 98. That was one of the golden era in tumor immunology, where really they appreciated how host-tumor interaction plays a major role in defining or editing the tumor, and the concept of immunosurveillance was well established. Although it was known from hundreds of years, but in science everybody asked for evidence. And at that time there were a lot of these genetic engineered mouse models were available, where we can knock out a specific immune system and can define that immune system is very much important for preventing incidence of tumor and also limiting the tumor growth. And those studies were all done in 90s, late 90s. Early 2000, they were getting translated into developing new therapeutics or ways how we can utilize them to take advantage of what we know in the basic science and make it more and more applicable. 2010, the first immunotherapeutics in many ways which was approved. This was like a vaccine. It's Sipuleucel, okay? Trust me, these companies, they always name these drugs so difficult, okay, that only they can pronounce it, okay, or they will hire somebody to pronounce it. And there is a reason behind it, okay? Do you know the reason why? If the name is simple, it's not important, okay? So they have to make it difficult, so you will think that it's a really, really good one. And honestly, okay, I have been to those meetings where they do these brandings, and of course, there are some of the initials of the people's name who is also in there, okay. But at the end of the day, they just want to make it so complicated, so people will think it's really, really good, because we even cannot, as a common person, we cannot pronounce their name, okay. So basically, this was the first tumor vaccine which was designed, and it was dendritic cells, which was ex vivo expanded. Again, we have given them the right antigen and put them back into the patient, and this was for prostate cancer, hormone refractory prostate cancer. And I told you, I think yesterday, that this company is bankrupt, because they don't have enough funding or support to continue, and insurance companies are not ready to pay, because this is in prostate cancer. And most often, when you have these castration-resistant prostate cancer, those individuals are over 75, okay, and they have already gone through surgery and radiations, and then they get this, and outcome we are looking to 2 to 3 months. So many patients, they have decided that now they have lived their life, good enough. They are in 75, 80s, in the late in the system, and they say, okay, they don't want to take it. So there is the audience, when our major role, player here. The first, immune checkpoint blocker was approved in 2011, the CTLA-4 targeting antibody, which we discussed yesterday. And then came the whole wave of PD-1 and PD-L1, antibodies, which were actually, if you see this is what I want to bring this into. CTLA-4 was approved for melanoma, and then came the PD-1, okay? And in PD-1, if you look at what they get approval for, so they got PD-1 approved for, a very specific, population in melanoma. Those basically which were unresectable metastatic melanoma and disease progression followed by the CTLA-4 antibody. And those patients who were resistant and non-responsive to CTLA-4 antibody, they got this one. Okay, so they have to find this special population where they can show the efficacy and show that this is this is a good one. The same thing goes for this another PD-1 antibody which was approved, and this is another PD-1 antibody. So there are multiple PD-1 antibodies from different companies which got approval, between 2011 to 2018 actually. And all these approvals, they get approval for the same antibody because they make it differently, and they target it to the different cancer, a population of patients, where they, and the targets basically is very selective. They want to show the best response. They will not just go into the general population or try to treat lung cancer, and show that it will respond. So they will go in a very narrow window and show that this will be effective, and then get the approval, and then they will go and clinicians can use it for anything. And PD-L1 antibody, first one was approved in 2017, and this was for urethral carcinomas, advanced urethral carcinomas. And another PD-1 antibody was also approved in 2017 for Merkel cell carcinoma. Another PD-L1 antibody was approved in May of 2017, and this was for metastatic urethral carcinoma. So it seems like urethral carcinoma was a pretty good target for PD-L1 targeting. And do you know why? This cancer was commonly used in all these three antibody clinical trials because they express PD-L1. Okay? So this was one tumor population, they were finding higher expression of PD-L1. That's why they used in all the clinical trials. These are all developed by different companies, and they are currently available in market. Other than immune checkpoint blockades or inhibitors, interferon alpha has been in market for a long, long time, and their patent expired in 2016, 2008, 2007. And what these companies usually do is that once patent expires, they will try to reformulate the same material into some other form, apply for another patent and get the patent, and then you have a new drug. So this is basically what they do is that they use pegylated interferon alpha, and the basic idea was that it will have a longer half-life and you don't have to give multiple injections. So pegylation is one way, many cytokines have been pegylated. GCSF, GM-CSF is also available now. This is an oncolytic virus which was approved by FDA in 2015. This was used for melanoma. And this one was the first CAR T cell. I don't even want to try to pronounce the name, okay? CISAGENLECL. And at the here. So all those all those monoclonal antibody, you got MAB, right? MAB. All these CAR T cell, you got this cell, okay? All of them will have this cell, okay? This is the nomenclature they are going into. Don't even try to remember what it is, but what it is that it is a chimeric antigen receptor for CD19, approved in 2017, and another one in the same class was approved in 2017 itself for B-cell lymphoma, relapsing B-cell lymphoma. Only difference is that these are from two different companies, okay? But they are both targeting the same, CD19. And many of them are right now in clinical trials, and you can see probably the list will keep growing every year because this area, as I said, is very hot. There are a lot of money to make. So if you look at overall, research and development activities in pharmaceutical company, almost half of the funding or support is going for immunotherapeutics in cancer, tumor immunotherapeutics. So if you have expertise, tumor immunologists right now are really hot commodity, and most of them are going to pharmaceutical companies because that's where jobs are and that's where every company is moving into. In all these, days, it leads to some real, interesting findings and achievements in many ways. Of course, in last 20 years, the major impact has been made, and most of them are, basically around this tumor microenvironment. Now we understand that microenvironment plays a major role. In 90s, like 1990, people were arguing all the time, no, it's all tumor cells. There is a nice documentary, if you go and look into, it's called Melodies of All. Yeah, it's a Siddharth by Siddharth. Right. Siddharth Chatterjee has written a book, and PBS, in a public broadcasting system in USA, they have made a documentary. It's a 6 hours documentary on cancer. Melodies of All, Emperors of All Melodies. And it's a 6 hours documentary, and I will highly recommend you guys to watch this. And then you will understand cancer really well and how it has evolved. At least our understanding has evolved, how much we know today and what we knew 30 years ago. This is not a long time. They cover Second World War. After Second World War, most of the research, what has been done in cancer research. I will highly recommend that go and either read the book. If you don't like reading, just go and watch this video. It's available on PBS, and I'm sure it will be on YouTube too. Emperor of All Melodies. Identification of gut microbiota as a major determinant of sensitivity for cancer patient in multiple treatments. It is going to be interesting area, and mainly it is happening for these immune checkpoint inhibitors. They are using it because here you are trying to manipulate the host response, and microbiota is turning out to be one of the major player in that context. The discovery that novel antigens expressed on cancer cell as a consequence of non-synonymous mutations. This is what I was talking about yesterday about those personalized, vaccines. These non-synonymous mutations are those mutations which are non-productive, functionally non-productive. They are not going to affect the biology of that protein, but it basically creates these new antigens, which can be, used for vaccine development and can be identified by comparing those tumors to the normal tissue. elucidation of innate immune mechanisms that provide major contribution to both natural and therapy elicited immune surveillance. Innate immune system is becoming more and more relevant, and there are a lot of new therapies which are coming, and most of them are like trying to induce actual really optimal level of danger signals. So there are a lot of different danger signals which they are utilizing to make it more optimal. And the identification of course, this is another hot area in cancer biology called exosomes. Exosomes are basically these extracellular vesicles which are secreted by the cancer cells and can serve as a vehicle to deliver antigen to different, or can be used for their advantage to create a metastatic niche. So exosome is a very important area in that. So these are all the translational advantages which have happened in last 20 years. And some of the technological advances have also been done in, at least in the area of tumor immunology, and that has been exploited. Definitely these, cost-effective platform for single-cell DNA and RNA sequencing. Immune system is very complex, and each cell within immune system plays a very different role. So defining, those single-cell analysis is very, very interesting and important, and this is all done because of these advancements which have been done. And of course, omics come into picture. Every omics you can think about is getting utilized. So there are a lot of activities going on trying to define and develop and design those robust algorithms, those softwares, and those other platforms to identify new antigens, new motifs, which will help immunology to move forward. And majority of these work are getting done in silico. So you don't need really a wet lab to do many of these experiments. Data are available, data sets are available, and you can utilize that. Refinement of CRISPR-Cas system for gene editing, this will come into picture someday. Someday we will engineer, everything, including human being, because that's what this system can do. They can basically create super babies, I don't know whether you saw that Chinese professor, he started, and his goal was to create babies which will be protected from HIV infection, okay, because there is one mutation which protects these things. Is called CCR5, which is a co-receptor for HIV. So if you can insert this mutation in any new baby, that baby basically will be protected from HIV. So that Chinese professor tried to do it. He got fired eventually because there are a lot of ethical issues are concerned, at this moment. And the introduction of mass cytometry and cyber sorting, this is another technology which are getting very popularized. That now nobody wants to do flow cytometry because it is tedious. I don't know how many of you have done it, flow cytometry. So basically, you create a lysate, you will send it to your facility. They will do this mass spec analysis, all sophisticated one, and then you can create what cells are there within that population, and you will have what you are getting from flow cytometry. You will get it from MS data. And same thing for cyber sorting. You are doing this expression profiling, and then use that expression profiling data to create the cellular population, what phenotype, what markers are expressed within that tumor or within that, patient population. So all in all, those are the technological advancement. And I think now, by this time, you guys are expert now in tumor immunology. But I think I have something to show you for basic research, okay? And this is a movie. You can read yourself, okay? I'm not going to read anything. And you will understand what it is, okay? How a paradigm is formed, okay? First of all, what is paradigm? Do you know what is paradigm? Whatever I said, you are going to follow it. That's called paradigm, okay? So paradigm is basically going through and following whatever is pre-existing knowledge or information which is given to you, okay? And whenever they are looking into that innovation in your grant proposal, okay, when you write proposals, you are going to write these innovative proposals. So first thing they will consider is that, are you breaking the paradigm, okay? Are you deviating from the conventional wisdom? And that's what your research is all about. Your research is not just to go and reproduce what has been done before. Your work is to just go and do something, find something which has not been done, or maybe if it has been done, try to prove it wrong. And before you prove it wrong, you have to know what it is, okay? So try to learn before you go and try to break it, okay? But my PC goes down again here. Sorry. Anyway. Yeah, it restarted. Anyways. So basically, this there are a few slides I'm going to show you what exactly paradigm means, okay? And you will understand many of us are in that same boat, okay? Especially those who are working in the same area, we all get attached, are emotionally connected to our own hypothesis, okay? And that's why the graduate students are important, because they have not been primed, so they can bring something new. They will say, hey, yeah, whatever Dr. Pai is doing is not right. Whatever she says is, maybe already been done, or it's not important. So I think that's where your thought process has to be, that to try to find something, which is novel, and that novel aspect comes if you understand, what has already been pre-existed. Yeah. Okay. So here, a group of scientists played 5 monkeys in a case, and in the middle, a ladder was put with the banana on top, okay? So there are 5 monkeys here, and there is a banana here. Every time a monkey went up the ladder, the scientists soaked the rest of the monkey with the cold water, okay? Not this monkey, but rest of the monkey are getting doused with the cold water. After a while, every time a monkey went up the ladder, the others beat up the one on the ladder. And why, you know that, okay? After some time, no monkey dared to go up the ladder regardless of the temptation, because they all knew that they have never been exposed, actually, that some of them have not seen that water. But they tell everybody not to go up because, as soon as you go up, water will come down. Scientists then decided to substitute one of the monkey. The first thing this monkey did was to go up in the ladder, because, monkeys, they love banana. Immediately the other monkeys beat him up, because they have already been, told that this is what happens. After several beatings, the new member learned not to climb the ladder, even though never knew why. This is a key word, never knew why, okay? And many of us do that, okay? When don't know why it is like this, but we will say, no, this is the way it is, okay? And that's what the paradigm is all about, actually. A second monkey was substituted, and the same occurred. The first monkey participated in the beating of second monkey. A third monkey was changed, and the same was repeated. Fourth monkey was substituted and being beaten, and the fifth monkey was also replaced. So now there is a pool of monkey who has never seen that water ever in their life, but they have experienced the beating, okay? And they don't know why they get beaten up. So what was left was a group of 5 monkeys that, even though never received a cold shower, continued to beat up any monkey who attempted to climb the ladder. Okay? And that's a reality. That's a reality what happens even in science today. So if it was possible to ask the monkey why they would beat up all those who attempted to go up the ladder, I bet you the answer would be, I don't know. That's how things are done around here. So that's what the paradigm is all about, okay? So many things we do, as a scientist, and at least when we are doing our PhD, we just do it because that's how it has been done. We don't even think about it, why we are doing it this way. And actually, this is where you have to develop that skill, to solve the problem. If you don't know why you are doing it this way, you will never solve the problem, okay? And if something happens, you get not the right results, you will say, oh, this is what I got. But you cannot go and solve, what went wrong. So I think you all have to start learning that. don't miss the opportunity to share this with everybody, okay? So go and talk about it. This is what happens, and we are all like monkeys, okay, in this. Thank you all. Only two things are infinite: the universe, human stupidity, and I'm not so sure about the former. That's our very famous scientist. And I think all I can tell you is that there was another lecture at American Society of Cell Biology, a famous scientist, her name is Professor Minna. And what she said is very interesting. As when we are getting you guys to go through the PhD, we are trying to make you guys more and more stupid. Okay? Because think about it, when you join a lab, whenever you have question, what answer you get? I don't know. You go and ask your senior some problem, they will say, I don't know. Your professor will say, I don't know. So you are among these people who really don't know what you are doing. And that's true, because you are going to work on something which nobody has done it before, right? So that's the right answer, that you don't know. Nobody knows exactly, what the answer will be. And you will have to go and find the answer. So by that time, by third year or fourth year, you will feel yourself really stupid. And I can tell you that by fifth year or third year, whenever you are graduating, most often your answer will be, I don't know, okay? The question, I don't know. So to me, I think that's the right answer. Whenever I say, I don't know, most of the time to my student, just go and figure it out yourself. Because really, I don't know. Anyway, it was really nice, and I enjoyed it, and I hope you learned something. And that's it. You should be ready for your exam, and good luck. Thank you. Yes. Okay. इसे फ्रीज हो गया लगता है. If you need picture, I can send you. Oh, okay. Oh. Yeah, do you guys have any questions?